Let's go ahead and get started. We have one talk today in seminar, and it is my pleasure to introduce my student, Amit Adhikari, who is joining us from a background in agronomy and soil science from Nepal. And today he's going to be giving us his PhD proposal seminar, and in his PhD he will be looking at assessing drought and grazing resistance through phenology, productivity, and carbon flux in C3 and C4 dominated grasslands.
Thank you, Dr. Jose. Hello and good morning, everyone. Today I'm going to talk about the grassland and their sensitivity towards different kind of disturbance. Let me begin with providing the basic overview of grassland. More than 1/3 of the Earth's terrestrial area is being covered by the grassland. Whether they are the steppes from Asia or rangeland from Australia or even the prairie from North America, all they provide numerous ecological services. They provide the habitat for wildlife. They sequester the atmospheric carbon. At the same time, they provide the support for livestock production. However, these kind of services being provided by the grassland varies mainly due to the grassland community composition. It is the grassland community composition that helps to maintain the structure as well as the function of the ecosystem. When we look at the grassland of the Great Plains, we can see the northern area of the Great Plains is mostly dominated by C3 species, whereas the southern area is mostly dominated by C4 grassland species, especially in the western region. This is mainly due to the variation, natural variation, which is present in terms of gradient from east to west in terms of rainfall, and from north to south in terms of temperature. Apart from this natural variation, the shift in community composition can be brought by different kind of disturbance, and among these disturbance, grazing and drought are two important factors which changes the species composition. There are several literatures which are supporting the fact that actually the intensive or excessive grazing is causing the shift in the grassland community composition from perennial to annual, at the same time from tall grass to short grass. Apart from that, there is one long-term research going on in northern Great Plains, and within the span of 80 years, due to intensive grazing, the grassland, the relative cover of the C4 community increased from 15 to 80%, which suggests that grazing is one of the important factors which affect the species composition. In addition to grazing, drought is also another important factor which has influence on the community composition. For this, we can look at the example of Dust Bowl event during 1930s, which was mainly due to the consequences of the severe drought. At that time, the grassland community composition shifted within hundreds of miles. So grassland community composition can be affected by the drought. So it will be quite interesting to know how these different grassland community provide the resistance and resilience for the drought as well as the grazing. And we can always measure the sensitiveness of this kind of grassland community in terms of their resistance as well as resilience. The resistance is the ability of community to maintain its structure and functioning at the time of drought, whereas the resilience is the ability of community to return back to its original state after the drought event has been finished. So resistance and resilience are two important key characteristics which provide the stability to particular grassland. Having said that, again, it is the community composition of the grassland because of which the resistance and resilience provided by various grassland varies. So, which brings the main objective of our research. They are assessing the resistance and resilience provided by different grassland communities towards drought and grazing. The second one is quantifying carbon flux in grassland communities during wetting and drying cycle. To address our objective, we'll be conducting the research in Cottonwood Range and Livestock Research Station, which is located in Jackson County of South Dakota, approximately around 100 miles east of Rapid City. The climate over there is continental, semi-arid, with 415 millimeter of average annual precipitation, and out of that 415 millimeter, more than 70% precipitation occurs during the growing season, suggesting the fact that the community composition, the structure and functioning of the community over there is particularly influenced by the precipitation regime. In the figure, we can see plot number 1 through 6. The plot number 1, 2, and 3 are representing the short grass, mixed grass, and mid grass. And plot number 4, 5, 6 are the replication of plot number 1, 2, and 3. And we will be conducting our experiment in these different plots. We have selected the Cottonwood Range and Livestock Station mainly because we can see existence of 3 different grassland communities. One is the mid grass, another is mixed grass, and the next one is short grass, existing in very similar kind of precipitation as well as soil regime. So the existence of these 3 different grassland communities on the same climatic regime will provide us with a unique opportunity to address our research question. We will be able to quantify the resistance and resilience or the sensitiveness of these different grassland communities towards the different kind of disturbance like the drought and grazing. So I believe this is the really good opportunity to understand about the role of grassland community composition towards their sensitiveness towards their disturbance. And the 3 grassland communities were achieved by the seasonal-long differential grazing management, which has been started from 1945 in Cottonwood. And 3 different dominated grassland species are buffalo grass and blue grama, both of them are C4, and western wheatgrass, which is C3. So the first research question for my research will be how do the waiting drying period as well as grazing and the interaction of both influences the C3, C4 and C4 co-dominated grassland communities. And to address the first research question, we will be doing a greenhouse experiment here in South Dakota State University. It will be a 3-factorial experiment in which the first factor is 3 different grass communities which will be achieved by using 3 different grass species, the western wheatgrass, buffalo grass and blue grama. We are particularly interested in using these 3 different grasses mainly because these 3 grasses made more than 90% of the biomass of Cottonwood Range and Livestock Research Station. So it will be interesting to understand or get the mechanistic idea how they will be affected by the drought as well as grazing. The second factor is the drought stress. For that, we will set up one control. In control, we will be maintaining the moisture level of 20% throughout the experiment, at least 20% throughout the experiment. And for the moisture stress, we will be maintaining the moisture level of 5 to 10% for 15 consecutive days before it will be watered again. The third factor will be clipping, which will do the simulation of grazing. The first treatment will be no clipping. Similarly, to simulate the wet period grazing, we will be doing the wet period clipping 5 days after water application. And to simulate the dry period clipping, we will be doing the clipping 10 days after water application. So altogether, there will be 18 treatments which will be replicated 10 times, and so the total number of pots we will be using is 180. So we will be using various response variables to measure the sensitiveness of resistance and resilience. They are the biomass, both below ground and above ground. We will be measuring the tillers, stomatal conductance, photosynthesis, as well as leaf water potential. Here, to quantify the resistance and resilience, we'll be looking at the biomass as well as gas exchange before, during, and after the drought event, as well as during the start and end of the growing season. And data analysis will be done in R programming language using linear mixed models. So the second question for my research is, how does the prolonged period of spring as well as summer drought influence the phenology and above ground productivity of C3 and C4 plant communities? Here the hypothesis behind this question is that the C3 plant starts their growth early in the season, because of which we believe that C3 dominated grassland species will be particularly sensitive towards the spring drought, whereas since the C4 grass start their growth later in the season, probably in the late spring or early in the summer, so we believe it will be more sensitive towards the summer precipitations or summer drought. So we are also looking at the phenology. We know that biomass gives the basic ideas about the resistance and resilience, especially in terms of productivity. However, to collect the biomass, it is labor intensive, it consumes a lot of time, and sometimes it might be difficult to achieve the higher spatial resolution. However, the phenology will provide us with the unique opportunity in terms of in terms of change in the start of the growing season, end of the season, and if there is any change during the peak greenness, that can be captured by using phenology. In addition to this, the phenology is derived from the remote sensing data, and because of which it has a greater extent, and it will provide us with the opportunity to address our research question, looking at the greater variations in terms of temporal as well as spatial resolution. So we'll be looking at two response variables, phenology and webground net primary productivity. To address the phenology, we'll be looking at the normalized difference vegetation index NDVI. It is actually the measure of greenness. In the figure, we can see that the vegetation which is green is actually absorbing the more amount of red light, because of which the NDVI value is high, whereas the vegetation which is dry or which is unhealthy absorbs less amount of red light, because of which the NDVI value will be low. And to calculate the NDVI, we will be using the satellite data from Landsat 5, 7 and 8. The Landsat actually have the spatial resolution of 30 meters, because of which we are using that. And another reason is we were thinking of using the MODIS, but the MODIS data is available from 1999, 2000 only, so it will not address our question. So we will be using the Landsat. So the first thing for this research we have to do is to define the drought period. We have to define the drought spring as well as drought summer. And to define the drought spring, we will be looking at the 3 or 4 years of spring precipitation, more 1 standard deviation less than the normal precipitation spring year. Similarly, to define the drought summer, we will be looking at the 3 or 4 consecutive year of summer with precipitation less than 1 standard deviation of the mean. In this way, we will be defining the drought spring as well as drought summer. After defining the drought spring and summer, our next step will be to define the resistance and resilience. For that, first of all, we will find out the time integrated NDVI from 1982 to 2016. Then we will be finding the NDVI for the years with dry summer or spring, as well as the NDVI immediately after the dry summer or the spring. So the magnitude of difference between the time integrated NDVI with average NDVI for the dry period will provide us the resistance, whereas the magnitude of difference between the time integrated NDVI with that of NDVI immediately after dry summer will provide us with the resilience. So in this way, we can find the resistance and resilience. Similarly, the resistance and resilience will be quantified in terms of shift in the peak greenness period, as well as shift in the start of the growing season as well as end of the growing season. So we can quantify the resistance and resilience and find the sensitiveness of these different grassland communities. In addition to phenology, we will also be looking at the carbon flux during the drought and wetting cycle. So we are interested to know how do wetting and drying cycle influence the carbon flux in C3 and C4 dominated grassland communities. So the carbon flux will be measured by using 80 covariance technique, in which we will be looking at the covariance of vertical wind speed with CO2 concentration. In the figure, we can see that the white big circle is representing the big eddies, whereas the small circle is representing the small eddies. These eddies are the parcel of the air which carries the CO2 in and out of the system. When they move up from the canopy, they carry the CO2 up, and when they move down, they mix with the air and they bring the CO2 into the canopy. And if there is flux of CO2 out from the system, if the flux is positive, then it is the carbon source, and if the flux is negative, or if there is more intake of carbon, then the community will serve as the carbon sink. So it will be interesting to know how these grassland communities behave as the carbon sink or source during the period of drought and waiting period. So this is the typical representation of the ED flux tower I am talking about. We are installing this tower which has got two special components. One is the three-dimensional sonic anemometer which will be used to measure the vertical wind speed, and the next one is open path gas analyzer which measures the CO2 concentration. We'll be installing these two ED flux tower, one at C3 dominated grassland communities and one at C4 dominated grassland communities as represented by the asterisk in plot number 1 and 3. And each tower will be equipped with one path gas analyzer, sonic anemometer, as well as we will be measuring the temperature, soil moisture, as well as humidity with using various different kind of sensors. Each flux tower will be installed at the height of 3 meters, so we can catch the CO2 coming from the larger distance, probably from 250 to 300 meters. For the data collection and processing, we will be using the raw flux measurement at the rate of 10 Hz. We are using the 10 Hz because the small EDs has the higher frequency, and it is not a good idea to measure above the 10 Hz because we cannot catch all those EDs or CO2 flux without using the 1/10th of second or 10 Hz. So we will be measuring the raw flux for 3 consecutive growing seasons starting from May of 2017. We will be recording the soil moisture continuously at that time, and we will be recording the ground biomass as well as leaf area at bi-weekly interval. All these data from the different sensors will be stored at data logger CR6 basics, and we have to deal with the processing and correction of the data we obtained from the carbon flux tower. And for that, we will be using the ED Pro software, which is developed by Lycor. We have to do the processing because there might be some kind of error because of unlabeled land, unlabeled sonic anemometer, time lag. There might be the gap in the data collection. So all these kind of things should be processed using ED Pro software. And we'll be measuring the resistance and resilience in terms of the ability of these grassland to come back to the stage of carbon sink or carbon source immediately after the period of drought or the wet cycle. So through this research, we'll have the basic ideas about our mechanistic ideas about the physiological response and functional response of these different dominated grassland communities towards the drought and grazing. And we believe, as far as I'm concerned, this will be the first research in which we are measuring the carbon flux in 2 different grassland communities which are coexisting in similar kind of environment. And I believe this will provide us with the unique opportunity to address the different issue related to the grassland community, and it will help us to serve as an important tool to develop the rangeland management strategies. Overall, this research will provide the basic understanding about the role of grassland community composition towards carbon storage, because any kind of carbon flux, or whether that is the phenological change in that grassland community, is mainly due to the community species composition of that particular grassland community or change in the soil by that grassland community. Finally, I would like to acknowledge my committee members. I would like to acknowledge South Dakota State University and Department of Natural Resource Management and my funding organization, Agricultural Research Station. With that, I am happy to take any further questions. Thank you.
Do any of the 3 species that you're working with exhibit phenotypic plasticity, where their morphology would differ according to environmental conditions?
I'm not entirely sure about that one. Yeah. but the three grass species exist in all those three different grassland communities, and I believe there is some kind of plasticity provided by these different grassland communities, but I'm not entirely sure about that one.
Your research, so well designed. So I really like that. So, two quick questions. You have a research experiment stations somewhere south from here. You have the same soil type and same precipitation, right? Same soil type, same precipitations. Why there are three different grass communities? We assume that if the soil is same, rainfall is same, vegetation should be same, right? That's the general ecology.
Okay, thank you for the question. The question was why there is three different grassland communities existing in same climatic regions, and probably I haven't described that one well. Okay, so there is a long-term seasonal long grazing experiment going over there. The animal are stuck at variable rate. So because of which, starting from 1945, and because of which there exist the three distinct grassland communities, because of stocking rate of animal is maintained differently in those three communities. And after once the three grassland communities target was achieved, now the idea is just to maintain those three grassland communities, because of which there is distinction between those three grasses communities. And yes, like you said, it will provide us the good idea about how sensitive they are towards the future climatic scenario, as well as the drought, as well as grazing.
Another quick question, sir. So you will measure the carbon flux, right? This carbon flux from the grass communities or whole ecosystem of that area?
So we are installing the ET covariance tower at the height of 3 meters.
Uh-huh.
And the carbon flux fetching distance for the ET flux tower will be 100 times of that height. So it will be around 300 meters.
Oh, okay.
And the one grassland community will be has the width of probably around 300 to 350 meters.
Uh-huh.
So we are pretty sure it will be that collecting that carbon flux from the same plot only, not from other. It will not be influenced by the carbon flux
outside
of other plot.
Question about those flux towers and just kind of the way that data is recorded. So a greater swirl within the eddy would raise that measurement, is that correct?
Oh, okay. So here the eddies are actually the parts of the year, and the larger eddies has the lower frequency, and they are generally found in the upper region of that canopy, whereas the small eddy are move with the higher frequency, and they are generally found in the lower region of the canopy. But it is not hard and fast rule like the larger will be in the upper and the lower will be in the lower region. There will be mix of the larger and upper, and they will carry that carbon with them, and that open gas analyzer will measure that carbon.
Okay, so,
So
more,
okay.
so more eddy flow
Yeah, more
makes
eddy.
more carbon available
Yeah,
to the sensor.
yeah.
So how are you correcting for potential influences of topography? Because that'll be more turbulent flow and could skew your measurements.
Okay, so the question was how to correct the topography. That is another good question. So the first thing we have to do is find out the flat area over there in different grass communities. The first basic idea will be to find out the flat area. The second is the measurement of carbon flux will be affected by the position of or orientation of these flux tower we will be installing. We have to orient those flux tower towards the prevailing wind direction. So we have to take account of that. We have to find the flat terrain or flat area, and we have to install that ED tower towards the prevailing wind direction. So which means if the wind direction is from one side, it is not necessary to install the flux tower right in the center of that plot, that grassland community. We can install in the side of the grassland community, provided that it will capture the carbon flux. Yes. So that will be the challenge for us to find out the flat area, to negotiate with that topographical feature. Yeah.
first research question, I think you talked about your experiment will be carried out on the greenhouse.
Yeah.
So my question is, are the greenhouse stuff from seeds or stuff transplant?
Okay. The question was whether I will be using the seed stock or using the live vegetation for the greenhouse experiment. Thank you, Dr. Silvan. So right now our plan is to use the seed, just to grow the seed and then transplant them. But we are also thinking about using the vegetative plant, bringing from the cottonwood. Since we will be using the soil from the cottonwood in those pots, it will be the good idea to simulate the scenario in the cottonwood to understand what we are talking about. We'll be doing some kind of pilot project before figuring out which will be the best method, because there might be a difference in the growth pattern if we are using the seed. Some plant may germinate earlier or some plant may germinate later. So we'll be doing some kind of pilot research before figuring out which will be the best appropriate method to go for.
Okay, the follow that question, you may talk about, I missed, how long in that greenhouse experiment, when you apply clipping treatment for the vertical grazing?
Okay.
And how much height, stable height you're remaining?
So
Maybe you're already on the time limit, I missed it.
So we are planning to start our experiment in the April or May, and we'll be clipping the plant. So the idea is like we'll be allowing the plant the 1 month time for their establishment. After that, we'll be initiating the drought treatment. And after the initiation of drought treatment, the clipping will be, to simulate the dry bed grazing, the clipping will be done 10 days after the treatment of water. And to simulate the wet bed grazing, the clipping will be done 5 days after the water application. The clipping will be done at the height. The clipping will be done to keep the biomass around 50%.
So you have a repeat clipping?
Yeah, repeat clipping each cycle. Like we'll be watering every 15 days after maintaining that 5 to 10% moisture level. We'll be maintaining 5 to 10% volumetric moisture level in those pots for 15 consecutive days, and then it will be watered again. And we'll be clipping based on that waiting and drying cycle.
You have to have the weight and the height relationship on it. You have to do it on that model before you know how much height you remove. You know what percentage of biomass to remove, 50% or 60%. And then you go next year into dormant and go next year, redo it again, or just one growing season?
Not just on one week.
Thank you. And do you have another question if you allow?
Okay, yeah.
I'm very interested in your second and third questions. I think you'll be down in the field, right? So you're looking at a pretty long period of spring and summer drought. What would happen in 2018, 2019? Then also, well, a couple of years going that way.
That's a very good question. Yeah. So the initial idea was to look at that summer drought influence, but now we are thinking of more looking towards the waiting and drying cycle. So you are talking about carbon flux measurement, I believe.
For both green up too, you know, your second and third, you all depend on your
Okay.
field, right? In the cotton field.
For the second one, we'll be actually looking at the historical data. We have the biomass data, and we have the weather data, and we'll be getting the phenological data from the remote sense data, and we'll be using the historical data from 1982 to 2016. So we have all those data,
Okay, I
and we'll be
see.
analyzing that data for the second one.
Okay, I see.
And we have looked at the data, and there is some kind of drought events going on
Yeah,
in the
right.
summer as well as spring.
Yeah, the historical
Yeah,
doesn't have
yeah,
drought in there.
yeah.
But what about the third one?
For the third one, we'll be looking at the drought and the wet cycle. So since the measurement is taking at the rapid phase each day and every time, So there will be the change in the carbon flux during the drought event, as well as there will be the change in the carbon flux during the waiting period. There will be definitely shift in that drought and waiting cycle. If not, there will be, if there is not like big drought events, there will be actually the shift in that drying and waiting period, and we'll be looking at the difference in the carbon flux between those three grassland communities during waiting and drying period, and we'll measure we'll quantify the sensitiveness in terms of resistance and resilience provided by those grasslands.
We should see if there's any questions from the remote folks. Does anyone remotely have a question? Okay, doesn't look like we have any questions remotely. Are there any other questions from the audience here?
I want to go back to the greenhouse experiment for a minute after what Shulan brought up. And I was curious about when you set this up, how long it was going to go, because I can imagine if you start from seed, you're going to have a different relationship early on versus later in the life history stage. And I wondered if you thought about actually selectively harvesting to get the relationships at these different stages throughout the experiment, and maybe enlarging it a little so you could selectively harvest. I don't know what's feasible, but to me then you would get this relationship with biomass and what you're doing, and you would, because it could change throughout the study.
Yeah, that's right. It could change throughout the study, mainly because of the fact that the plant will be growing and the seedling stage or the early stage of the growth will be quite different from the later part of the growth. So that's why we are not like rigid in terms of maintaining that 5 to 10% moisture level for 15 days. There might be flexibility based on that how the plant will show the symptoms. If the plant is showing the wilting symptoms, then we'll adjust our watering events. That's another factor to be taken into account.
Okay, any other questions? All right, thank you everyone. Have a great spring break and we'll see you in 2 weeks.